So let's start with module 6.1, which is interface and F-phase. You're very familiar with this. So when we talk about cell cycle, of course you already know that this is leading to cell division and then production of two daughter cells, and the chromosomes will be very identical. So that will be the characteristics of the cell cycle. Now of course you already know that division or cell division is very important in order for organisms to grow or repair their wounds. So that's why cell division will be leading to these three important characteristics. Cell division, two daughter cells, and chromosomes will be identical. Now, the most important thing you have to take note about the cell cycle is that there are two major parts, which will be the interphase stage and the M phase. Starting with the interphase stage, you already know that these are divisions in between or in between divisions, and this is approximately 90% of the cell cycle spent by the cell in the interphase. So the interphase can be divided into phases, which we call as a G1 phase, the other one is the S phase, and the other one is a G2 phase. So let's just go to the most important characteristics of each of the phases. G1 is metabolically active, get bigger in size, DNA not yet replicated, and the organelles replicate. So of course it's very important that the size cytoplasm increase and the organelles replicate, so that this can be distributed equally to the two cells, so that one of the cell will die because of less cytoplasm or loss of organelles. S phase, the most important thing you have to take note is DNA synthesis or replication, and also the presence of production of the sister chromatid and duplication of the centrosome. So those are the most important things in S phase. For G2 phase, it is further cellular growth, and of course it double checks the duplicated chromosomes for error. And if there are errors, either the cell will go into apoptosis or will stop its division. Okay, so this will allow the cell to complete its preparation so that it will be able to do the M phase. So just take note that in the G0 phase, some of the cells like muscle cells and brain cells exit the cell cycle and do not divide anymore. So there are cells that are like that. So we discussed this already, so just take note that if G1 there are 50 picograms of DNA, since in S phase it will double, you will expect to have 100 picograms of DNA, and from then on it will have the same amount of picograms of DNA until at the end of M phase it will return to the original. So at least for the G2 phase, you will have 100 picograms of DNA. In terms of chromosomes, if there would be 21 chromosomes at G1 phase, you expect to have 22 chromosomes at S phase as well. So since the number of centromeres did not increase, you would expect to have the same chromosomes as well. For G2 phase, it is the same, 22 chromosomes too. Now for question 22 chromatids at G1, how many would their chromatids be present in S phase? Since there's a duplication of the sister chromatids, duplication of the DNA, you would expect times 2, so it becomes 44 chromatids, and it will enter G2 with 44 chromatids. Later at M phase, this will be separated at anaphase, and it will return to 22 chromosomes by the end of the telophase. Okay, so just take note of the important characteristic of each one. So for mitosis, stage of the cell, that cycle that occurs before mitosis, so the nuclear membrane is present, and then the DNA cannot be distinguished, or the chromosomes cannot be distinguished. And then for mitosis, prophase is very important here, wherein the DNA has already condensed into visible chromosomes, and the nuclear membrane, though present, is already starting to disintegrate. So that's important thing about prophase. For metaphase, most important thing would be that the chromosomes would be aligning at the center called the metaphase plate. Individual chromosomes will be on the metaphase plate. So you already know that in metaphase 1, this will be homologous chromosomes on the metaphase plate, but for mitosis, only individual chromosomes are exactly on the metaphase plate. In addition, we have the anaphase in mitosis, wherein the sister chromatids will be pulled apart by spindle fiber going towards the opposite side of the cell, and then telophase wherein the nuclear membrane has already formed and then your chromosomes are already beginning to unwind. Now, one of the most important things you have to take note would be the number of chromosomes. So if you have prophase 1, 2, 3, 4 chromosomes, you will expect in metaphase to be still 1, 2, 3, 4 chromosomes. 1, 2, 3, 4 chromosomes as well. But in anaphase, the chromosomes will separate its chromatids. So depending on the question, you will have different number of chromosomes. So for example here, per cell, since the chromatids have separated and you have now multiplied your functional centromere, so you have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes per cell in anaphase stage, and there will be 1, 2, 3, 4 chromosomes per pole at anaphase stage. Now at the telophase stage, you will now have here 1, 2, 3, 4 in this pole, 1, 2, 3, 4 in this pole, since they have not yet separated. So per cell, it will be 1, 2, 3, 4, 5, 6, 7, 8 chromosomes per cell. But once these are already separated from each other, although the picture is not here, then you will expect 1, 2, 3, 4 chromosomes in one cell and 4 chromosomes in the other cell when there is already complete cytokinesis. Alright, so again, this would be first picture is anaphase, the second picture is prophase, the third picture is telophase, the fourth picture is cytokinesis, the fifth picture is metaphase, and the last one would be prophase. Okay, now of course you've already answered this. So number 1 is prophase, number 2 is M-phase, number 3 would be the whole of this is the interphase stage, number 4 this is the anaphase stage, number 5 this is the S phase, number 6 G1 phase, number 7 is cytokinesis, number 8 is G2 phase, number 9 is metaphase, and number 10 would be telophase. So it's very familiar